series. My name is Matt DeCamp, and I'm on the faculty of the Sugarman Institute and in the Division of General Internal Medicine here at Johns Hopkins. In case you weren't aware, didn't receive a late notice, our original planned speaker, Eric Younks from the University of North Carolina at Chapel Hill, is actually stuck down there because of the recent winter storm. We hope he's warm and safe. But because the topic of gene editing is so timely and important, we put together a panel of our own to really help foster a discussion, which I'll coordinate. I'll serve as a moderator, but I want to start by introducing our panelists, and I'll tell you how the session will go. So Dr. Debra Mathews is the Assistant Director for Science Programs for the Johns Hopkins Berman Institute of Bioethics and an Associate Professor at the Department of Pediatrics. With a PhD in genetics, her work focuses on ethics and policy issues in biotechnology, with special interest in genetics and stem cell biology, including, of course, CRISPR, which we'll hear about today. Dr. Jeremy Sugarman is the Harvey M. Meyerhoff Professor of Bioethics and Medicine, again here at the Berman Institute. He's internationally known and has significant experience and expertise in diverse areas of research ethics, but here I highlight his work on HIV prevention research and research with emerging technologies such as stem cells. Third, Dr. Jeffrey Kahn is the Andreas C. Dracopoulos Director of the Johns Hopkins Berman Institute of Bioethics and Levy Professor of Bioethics and Health Policy. His work at the intersection of ethics and health policy is internationally known, and I'm sure we'll hear about his experience at the second International Summit on Gene Editing, which was just a few weeks ago in Hong Kong. So our plan will be as follows. First, Dr. Mathews will give us some scientific background on CRISPR. Then Dr. Kahn will talk about some of the important policy issues and recent developments in the policy landscape. And then third, Dr. Sugarman will introduce some of the ethics issues. After those short presentations, we'll have the panelists up at the front for audience Q&A. So I'll start with Dr. Mathews.
All right, thank you very much. So I'm just going to do a very brief introduction to the science of genetic modification. So in talking about genetic modification, there are multiple targets that we have, and of course multiple techniques that you can use. With regard to target DNA, you can be targeting the somatic DNA, so DNA in the cells of a person in front of you, usually a sick person in front of you, that you're trying to help with the disease. Germline DNA, which is the DNA that transmits genetic information from one generation to the next, is found in eggs, sperm, and embryos. And then mitochondrial DNA, which is sort of a separate thing, and we're going to leave to the side for today. With regard to methods or techniques, gene transfer or gene therapy, as it is also called, is modification targeting somatic tissues. And then gene editing, which is this new technology that we'll be discussing primarily today, that can be used either for somatic modifications or germline modifications. And again, I'm going to leave mitochondrial modifications and techniques to the side. On the somatic side, we've been modifying DNA for a very long time. The first clinical trials began in the early 1990s with adenosine deaminase deficiency in kids with severe combined immunodeficiency. This involves the delivery of a therapeutic transgene to treat, again, a sick patient in front of you. Historically, this has been done for single gene defects, but it's expanding, and there have been almost 500 clinical trials, and we have a method of oversight for this kind of gene transfer. The Recombinant DNA Advisory Committee, which has long looked, since the 1970s, looked at recombinant DNA research, now primarily focuses on gene transfer or gene therapy trials. But what we're talking about today is the new technologies for gene editing. So it's not just CRISPR, which is the one you hear the most about. It's also zinc finger nucleases and talons. And the reason CRISPR is so revolutionary is whereas prior technologies, well, CRISPR, talons, and zinc finger nucleases, the new gene editing technologies, are so revolutionary is because in the past, if you wanted to make a specific modification we didn't have good technologies to do it. So often what we had to do was sort of a scattershot approach where you modified lots of things, not in humans, in a simple model organism, and selected out those mutants that were interesting or did the thing you were interested in studying. What's different now is that we can be much, much more specific about this. So these new gene editing technologies, akin to Word, but without the annoying paperclip, for those of you who remember that, enables you to cut and paste really, really precisely, although it is not perfect. And then after you do the cut, you can repair that cut in several different ways, either to eliminate the functioning of a gene or to modify the functioning of a gene. As I mentioned already, there are several types. Zinc finger nucleases have been around for a long time. TALENs, also for a while. CRISPR is the newest, and it is also the least expensive and the technologically easiest to perform, which is are big reasons why it's sort of taken the field by storm. But again, these aren't completely new technologies. CRISPR is actually part of a prokaryotic adaptive immune system. So I'm not going to go into the details, but it allows bacteria to fight off infection from phage. So when a new phage infects it, it shreds its DNA, puts pieces of that in its own genome, and uses that to recall that infection later. And it's this immune system that has been co-opted to be used now in mammalian, including human cells. And as I already mentioned, there are a number of different ways to repair the mutate or the break that you make with this system. And it can be used somatically for in germline editing, for reverse genetics rather than forward genetics, where you identify a phenotype and then study. Reverse genetics, you can break something and see what happens. Gene therapy, this has already been done using CRISPR in a number of diseases. And then, of course, human germline genetic modification, which is what we're here to talk about today. Again, this technology is much, much more specific, but there are still problems with off-target effects, meaning maybe you get the thing you mean to modify, but then you also modify other things in the genome, and mosaicism, meaning you have a multi-celled thing that you're trying to modify, and you only modify successfully a percentage of those. This is getting much, much better very rapidly because of the number of people using this technology, but it is still a concern. And this is just a picture of Layla, a little girl who, in fact, benefited from the somatic use of this technology. And I will stop there, because my last slide is about FDA regulation, which I think you're going to cover. Maybe.
We can come back to that. So before I is it going? Yeah. So before I started, I want to acknowledge what we probably surmise is that we were scrambling over the weekend to figure out what to do about the snowstorm south of us, and Eric Young's inability to get here. And I wanted to say, Matt worked very hard over the weekend. He took time from his family to pull all this together. And the fact that we have so many people in the room, I think, is a testament to our being able to scramble. So it may have looked easy, but it was a lot of work on some people's parts. So thank you for that. I'm going to give a little bit of background about what's happened in the last couple of weeks, as Matt alluded. Jeremy will then say a few words about the ethical issues raised, and then we'll open it up for panel discussion and your Q&A. Okay, so this is, of course, the big news of the moment in the world of biotechnology oversight and CRISPR and how these technologies get used. So as you see there, the 25th of November, there was an article published in the MIT Technology Review, which was at that time truly an exclusive, that there had been an attempt, is what that story said, to create genetically modified children. The story then sort of got more interesting over the next few hours and days, and that actual babies had been born, these twins that supposedly exist as a function of the technology that Debra described being applied for the first time to human embryos that weren't implanted into a woman's body. So there had, as you probably have read if you've been paying attention to this story, Dr. He goes by J.K., much easier for Americans to say J.K. than to pronounce his surname appropriately. Detailed via YouTube, and then, as I'll say in a second, at the International Summit in Hong Kong, what he had done, how he had done it, being a little bit out of the typical way that research findings and successes are reported. Right? There had been no publication in a peer-reviewed academic journal. The first story first broke, as it says here, in MIT Tech Review, then the AP got a story published, and then there were these 5 YouTube videos, but still no academic publication, so no ability for the scientific community to assess what had been purportedly done. So, huge amount of media coverage as a result, and the timing, hard to imagine was coincidental, even though it's been described that way. In that, the reporting was on right at the outset of what was a convening of an international meeting. So the meeting began on the 27th, but the community of people who were attending that meeting, I was among those on the program. I spoke actually on the same day as Dr. He spoke, had been arriving in Hong Kong in the day or two before just because of the way the flights work. So pretty much everybody was in town when this news broke, meaning that the media didn't have to go far to find people to talk to, which is actually an interesting part of the story. So the media all sort of congregated in a single place where the summit was going to be held, first at the hotel where we were all staying, and then at the actual venue, which was the University of Hong Kong. And so they actually didn't know who they were speaking to, but they figured if you were there and had a name tag on, you were probably an important person, and they stuck a microphone in your face and asked you some questions. I'm not exaggerating at all. So there was a sort of media frenzy that was made easier by the way this all unfolded. This was the second of international summits that had been convened by international bodies. The first one was in December 2015. It was in Washington, D.C., and it was convened by the Royal Society, that's the UK, the 2 national academies here in the U.S., and rather than Academy of Sciences Hong Kong in 2018, it was the Chinese Academy of Sciences in 2015. It's a distinction we can talk about if you're interested. There are some important political differences between Hong Kong and its academy and the mainland. So the point of these summits, which are supposed to be 2 to 3 years apart, is to give the community an opportunity to allow the science to advance, talk to each other, and make, if not judgments, then at least talk about it and maybe come to something like a consensus about appropriate uses going forward. Sandwiched in between that meeting in 2015 and this one in 2018 was I want to go to this and come back, sorry, a report, an international consensus report, which was convened by the same bodies as the 2015 summit. I served on that international consensus committee, which issued a report which had, among other things, the criteria for how to go forward with genome editing that would create heritable modification. So that's the kind of genome editing that we are hearing reported. And the answer is there's a lot of criteria that have to be met. It has to be for deadly diseases that have no other option, and a lot of research on animals before you go forward in the first time in humans. So the point of showing this now is to say this was sort of the stepwise process that had been proposed. The second meeting in Hong Kong was meant to be a sort of follow-on to both the 2015 summit and this consensus committee report, which was laying out a very careful process. And then we got this. So J.K. Head came to the summit. He was actually on the program to give a talk about the state of the science. And then the news broke that not only had he been working in vitro in laboratories, which people in the CRISPR community knew about, that he was working with in vitro human embryos, trying to introduce CCR5 mutations. That was not news. What was news was that he had, during the time he was discussing and describing his bench work, had been actively implanting modified embryos into at least one woman's body. He was trying to get women pregnant. And he gave this presentation. It was actually at the time on the program that he was originally scheduled, but there was a sort of clearing of the program around him to create more opportunity for his presentation and discussion. There's a lot of drama, not only because there were so many media in the room, but because he disappeared from the hotel where we were all staying the day before. He left. Some people thought he left Hong Kong altogether, but he left because he was getting death threats. Went to some undisclosed location. He was not in a room that any of us saw at the time of the point on the program when he was due to speak, and so his name was announced, and there was a very, very long pause. Anybody who was watching on the webcast would have seen this, 45 seconds or so, and then a door opened to the side of the stage, and he came marching out. So it was a sort of a lot of drama. He presented slides which showed the first time what he purported to do, and then there was a Q&A, more like an interrogation, frankly, by two members of the planning committee of the summit. That's Robin Lovell-Badge on the left, and the back of the head of Matthew Porteous. Robin Lovell-Badge from the Crick Institute in London, and Matt Porteous from Stanford, who grilled him without a lot of clear answers coming back about why he did what he purported to do, and all sorts of questions about things like how many embryos were edited, how many women are there, how many women are pregnant, how many women have you tried to get pregnant, what was the consent process, and so forth. Okay, so his claims are, as you probably have already read and you heard Debra describe the tools. So he altered embryos from 7 couples receiving fertility treatments. The consent form that we've all seen is an English translation. So I don't have access to Mandarin, and I don't know anybody who has read it in Mandarin to give us a sense of whether the translation is accurate, but it's very unclear about whether the couples who were offered these services understood exactly what was offered to them. Of course, it's the paper, not what they were told, and all the things that we say about consent. As you see, there's a claim in his slide presentation that he went through these steps to make sure that the edits were as he hoped. As it says there, 16 of the 22 embryos were edited. He said during the presentation that there were actually 31 embryos, so there's already some mismatching in the numbers, and it's going to be hard to know what the facts are. And then it says 11 were used and 6 before the twin pregnancy was achieved. That's in the one couple, one pregnancy achieved so far. He's been very coy about whether there are others. The other thing to say is he has disappeared. No one knows where he is, and he's sort of fallen off of the public radar. The Ministry of Health in China, the mainland, has said that his work has been suspended. That's their word, and that laws have been violated. Chinese law has been violated. So not clear what's happened to him. The presentation named the girls, as you see here. We're not sure whether those are the sort of nicknames or code names or whatever kinds of names, but the 2 girls who were born and been given these names, they were born in November, as it says here, according to Dr. Head's presentation. And it's a little bit unclear about whether he actually achieved what he set out to do because of some of the scientific information that he presented. Now, none of this has been independently assessed. There's no way to know whether his slides were fully informative or accurate, for that matter. All right, so he did say in the course of the questioning, the Q&A part after his presentation, that there is another potential pregnancy. So it's not clear whether that means he's still trying or whether a woman is maybe pregnant and they're waiting to see. It's not clear what happens when the Ministry of Health in China says his work has been suspended, whether that means there So it's interesting, and I think Jeremy's going to raise this a bit in his comments. It's not entirely clear why you would do the mutation that he proposed. So CCR5 is a mutation that it purportedly would provide some immunity from HIV infection. What the reason for that is in this particular case is really difficult to understand, and he was quizzed about that. Probably that was the greatest level of detail that the back and forth got into. Why was this an appropriate thing to introduce to an individual? And he kept saying, because HIV and AIDS are a terrible public health problem, which is true. That second part is true. But why genome editing of embryos is a way to attack a terrible public health problem is very much unclear, and why in these particular children that was an appropriate thing to do. All 7 cases, all 7 couples apparently have an HIV-infected father, husband, and so that's part of how they were recruited. There are ways to use IVF and sperm washing to avoid HIV infection that don't require genome editing, and so it's not as if that's the only way to have a HIV-free offspring. It may be the way that those couples were recruited, and I guess we'll hopefully find that out at some point. So he claimed that there's a real world medical value. It's not at all clear to, I think, anybody who's thought about this, why that's the case. Here is the statement at the end of the summit, which a little bit unfortunately says two different things. This may be the one that was at the midpoint rather than at the end. So there were 2 statements issued, one sort of in the immediate aftermath of this story breaking, which is this statement, and then there was one at the end of the summit, which more or less say the same thing. They condemn what it says he did and point to the report that I showed you the screenshot of a second ago, along with a Nuffield Council report, which is the UK Standing Bioethics Commission, which issued a report in 2018, more or less coming to the same conclusions with a little bit of nuance, which we can talk about if you're interested, and saying this was irresponsible, inappropriate, violated international norms and values, and it should never have happened. Okay, I showed you that already. Maybe we'll switch gears now.
Jeff, can I ask for clarification?
You sure can. Let you ask with the microphone. This is Dave Valley who's asking the question.
Could you just clarify the alleged genotype of the two twins?
So we can go back. Is it that one?
No, the previous one. Okay. So, Lulu is either heterozygote or mosaic. Fair enough. Nana also appears to have normal copies of CCR5, somewhere less than half. But then down below you say Nana has 2 mutations roughly in the same part of the gene as a naturally occurring mutation. So I don't understand for Nana, is she homozygous for CCR5 mutations, or is she also ahead?
So this all comes from the slides that were presented in Hong Kong. And so, and now it's second-hand reportage of the slides in Hong Kong. We don't know, the short answer. There was a lot of scrambling and many of us were taking pictures of the slides during the presentation, which were not shared and have not been published. I was tweeting madly, trying to send out information in real time, as were many others, not just me, and there was lots of filming happening. I'm happy to share the slides with you, but there was a lot of consternation, a lot of concern about these kinds of questions. I mean, the scientific community is like, what in the world did you do? How did you do it? How do you know you did what you said you did, and so on. Among the things that the organizing committee of the summit called for was an independent international investigation to get to the bottom of this. It's going to be really hard to know whether that ever happens, frankly. Okay.
Great. So thanks to both of the prior speakers to sort of set the stage for this, and some of the points that we touched on. I should say that some of this is now available, so even though we don't have a scientific publication, which is what we really want to be able to understand what actually happened in this case, we can get close to understanding some of the things that happen by the materials that have been made available in sort of informal meetings, which raises some interesting questions about scientific publication. The transcript from his presentation is available on the web freely. The consent document translated into English is also available. There's some good coverage in all the major outlets, including 2 really great pieces in the New York Times that give us some sense of what's going on. And I also want to thank Matt and Alan for pulling together all of these slides that we've been using as well this morning. So there are multiple ethics and policy concerns that arise out of this, and I'm just going to touch on a few of them to just highlight those so we can discuss them. Every group that has deliberated about whether and when it might be appropriate to do any kind of germ modification has said that there has to be elaborate oversight. All research with human subjects globally there's an accepted standard that there's oversight. In this case, there was 0 oversight. And so without an independent review prospectively, it's hard to sort of get the science right. What's an interesting transition here is basic scientists may not be accustomed to having the same kind of oversight that people who are working in lab sciences or also working with human subjects have. The place that this has come to bear is with stem cell research, where we do have a stem cell oversight committee here at Hopkins and many parts of the world, in which it's an unusual thing for basic scientists to understand the necessary role of an oversight body. But the National Academy's report that came out and every other international body that's looked at this says, if and when it's time to get there, and no one has said yes, as Jeff said, there would need to be carefully articulated principles about how to go forward. So here's a clear evasion of the need for oversight. Both Deborah and Jeff touched on this off-target effects, the fact that although CRISPR is very precise, it's not perfect. It doesn't necessarily only go to the place that you want it to go. And in addition, you have this issue of mosaicism, which David was raising the question about what's actually happening in this case. Did we get all of the cells? How many of the cells? What happened in this case? We don't know, but we know enough to know that in the other times that this sort of work has been done in human embryos, and it's been done twice before and reported in 2 separate instances, we know that there were far more off-target effects than we anticipated. And those were done in more safe environments. We can talk about those experiments in more detail if we need to. The consent issue was interesting. The consent form that I saw, also the English version, not the Mandarin form, it's long, it's confusing, not a surprise. It could be a translation issue, but it begins to say that this project was an HIV vaccine project. Vaccine translates from Mandarin. I've seen it. It is not gene edited. So the question of how the consent process took place, through a variety of sort of media sources, there was some discussion, I think, in the transcript as well. He describes having some of the people in his lab, meeting with people for a while, and then he himself engaging in a more formalized consent process using the form. The question of knowing what these people consented to and how that was done is still unclear. Now, one huge science and ethics issue is this one about the target, what he was going after. So where did this come from? So just as background, the CCR5 receptor on the T cells in human beings confers, it's how HIV gets into the cells. If there's a delta 32 mutation of that receptor, the virus can't get in, and many people who have that naturally occurring mutation are relatively immune to HIV.
Homozygous.
Homozygous. Yeah. Homozygous. Yes. Thank you. Now, it's not surprising that he went there from a sort of scientific standpoint. There was a prior study that was reported in the New England Journal about 3 years ago. A group out of Boston published this paper in which they took the somatic cells of people living with HIV. They took those T cells out. They used TALEN, so one of the other forms of gene editing. They edited the CCR5 receptor. They reinfused those T cells into people with HIV. And they compared how long it took for those people who had now these edited cells in their body to have a viral rebound. So they were undergoing treatment with antiretroviral treatment. They didn't have detectable virus. Cells are taken out, mutated, put back. Those with mutated cells took longer to viral rebound than those without. That's an elegant study, and it shows, in a scientific way, the plausibility of editing the gene to have a somatic effect. But whether or not we would now go into the setting of the germline, where we don't necessarily have, as Deborah said, a sick patient in front of you, these would be presumed to be healthy, right? So if according to the consent document that is available on the web, sperm washing was done. The likelihood of these infants being born with HIV was low. If there was a thought that they would be born, there are lots of things that can be done in the perinatal period to decrease transmission. It's done globally all the time. It's called PMCT, prevention of mother-to-child transmission of HIV. You can give the mother antiretroviral treatments if she's infected. You can then treat the neonate, and you decrease the likelihood of perinatal transmission less than 8%. Had the child been born with HIV, it's possible to put those children on treatment right away and treat them. So there were multiple alternatives to intervening in this way. And this raised the question about what was being done here. If you're doing it as prevention, this wasn't what the national academies or any of the international bodies thought would be appropriate, that we would be doing it for diseases that lack other treatments, and we would be doing it for treatment, not necessarily prevention. Which raised the question about, was this tilting towards something else that people were deeply concerned about? Or are you trying to really enhance people instead of treat them for a disease? And how would you have a
I just
question?
wanted to ask for a quick clarification. You said that, so if I naturally had this particular mutation that made the Delta mutation,
CCR5.
I still can become HIV infected, but my course may be very different than someone who doesn't have the mutation.
So it's unclear because it's almost an unanswerable question. We know that people who have, right? Because in that setting, we don't know, because there may be people who haven't been exposed to HIV, and we're not going to do that experiment. But what we do know from CCR5, so thank you. So this receptor, and this is really critical in the HIV cure space, where people are trying to come up with cures for HIV infection instead of treatment of infection. So even though HIV is completely treatable, 1 pill a day if you have access to that pill, tolerated by many people, there's a hope for many people with different diseases. Could there be a cure? Whole space of HIV cure research driven by a test case, a guy named, originally called Berlin patient. Guess where he had his procedure done? Yeah, it's tough crowd at Hopkins, isn't it, right? So he's in Berlin, and he's living with HIV. He develops a malignancy. He needed a stem cell transplant. The doctor doing that stem cell transplant found someone who was a match who also had the CCR5 mutation. He received that transplant. He actually rebounded in terms of his malignancy, had a second transplant. He took himself off antiretroviral treatment. The doctors didn't recommend it, and he's been off for almost 10 years. So he is the first patient cured of HIV using a similar technique. Okay? But again, it's a different question to know how it would work in terms of prevention. So the specter of enhancement has been things that we understand, and maybe if we're going to make exceptions, we're going to do so for lethal diseases, debilitating diseases. But whether we want it to make people smarter or better looking or less inclined to be infected with disease is the second question. And the final piece that's received a lot of attention is the notion of follow-up. So if in fact you intervene truly in the germline, that means it's going to go to the next generation. So what's the right follow-up? And how do we know what those effects are going to be in, for you scientists, your F1s, 2s, 3s, those generations, right? What about your kids and grandkids? And how do we secure follow-up at ages where people are maybe likely to reproduce when they're not so careful, sort of like teenagers? Come on in. Let's check. Let's follow. How do we do that over time? And it raises some other interesting ethical questions about intergenerational consent, about whether it's appropriate to be able to provide that. So that's just touching on some of the issues. There are many more, but I'll just stop right there. And what we'll do is, three of us will come up to the table up there, and Matt will moderate the discussion. Since we're trying to make sure everybody hears, please wait for a mic to come to you. That will help us.
Great, thanks. And while the panel settles, we do have about 25 minutes for questions, so plenty of time for discussion and encourage. Many of you that raise your hands, we'll bring a microphone to you.
Deborah, I have a follow-up. So as I understand it, the technology is able to cut a piece out, and you might then be able to replace it with a better functioning gene, for example. But what does that mean when you're talking about more than one cell?
Right, so the technology is designed to go into all of the cells.
There are.
Right, but that's the problem with mosaicism, it doesn't always, right? Or maybe it gets in those cells but doesn't efficiently cut, doesn't do its job. So you end up with some percentage of cells that have the change, some percentage that don't. And in the case of the twins, given that they do both appear to be mosaics, their risk of HIV infection is going to depend on which cells ended up with the change. So,
Yeah, mucosal cells would be good.
right.
What I read in one of the newspaper reports that he worked for industry, not an academic institution, and he paid those ladies a lot of money.
So we're not sure, so I haven't seen that report, but one of the things that we knew is he had a research fund when he was working at the Southern University of Science and Technology. But he didn't do his research, these cases, under the aegis of the university, and he explicitly said that he didn't do that in that way. He did secure services for where the babies would be delivered and how they would be delivered, and that was separate from that university, which the same city, different place.
Although the consent form did say that his funding was from his university.
It's all really unclear. He also has two companies of his own and has applied for patent protection on some of the techniques. So it's complicated, I think, is the story, and we're hearing about it kind of in a backwards fashion, right? It's the opposite of transparent, and we're learning about it after the fact.
And apparently two scientists at Rice, who is where he did his training, right? Also, US scientists have a stake in his companies.
So to make matters even just a bit more interesting, while someone's reaching for the microphone, Holly Taylor forwarded me a stat news piece from today talking about another set of experiments that he conducted dealing with familial hypercholesterolemia, in which he also supposedly is doing similar work. So again, a preventable or treatable condition that you may argue on the same grounds. That the reason why the announcement came out is that the paper was not published by a scientific journal, and the reviewers were saying that it wasn't publishable. So it's an interesting piece, but here's a guy that's definitely renegade. And the other piece that came up is I was asked by an editor of a major science journal about what they should do in terms of considering or not considering an unethical piece of research. And this is a long-standing debate in the ethics literature about what do you do with unethical research. We all need those data to understand what happened. At the same time, if we reward that with a scientific publication, that doesn't send the right message. And so when ethicists often deliberate about these issues, they tend to come down on the side of publication with really stern spankings that go along with it, to be able to let the discussion come forward.
Well, if you recall, now almost 4 years ago, when the first genome-edited human embryo paper was published, right, the scientific community knew about it because it had been bounced from journal to journal to journal all winter, getting rejected. And it ended up in Protein and Cell, accepted or submitted on March 30th and accepted on April 1st. So not the highest impact journal that one might hope for. But this hasn't even gone through that process.
I mean, I think there's, as we say, there's much to sort of figure out here about moving forward. Maybe that's what we should spend a little time talking about. Sort of how does the community recover and move forward? So there's sort of a version of this story that this is a misstep by a renegade. Clearly not a charlatan, however. I mean, this is really interesting distinction, right? This is somebody who was part of the scientific community and was working on bench science that was disclosed to his peers. This bench science, what he didn't disclose to his peers, that he was implanting these edited embryos into women. So someone said, I would not want to play poker with this guy because he's got such an ability to keep secrets. And what does the scientific community do about a case like that, I think, is a really important question. And is it a one-off, or is this a problem that needs to be dealt with in some policy respect?
So with regard to renegade versus charlatan, though, I mean, there was a lot of lack of transparency, it appears, with the people he was recruiting.
Yes, but I don't think he was selling something like snake oil.
Well, yeah.
He knew he could actually do the things he was saying he was
Well,
doing.
he
The
could maybe.
science part, the science part.
So I was kind of curious because so three of you have each referred to the scientific community in their response to that. But as we all know in this room, the scientific community isn't a monolith, right? So I was wondering how you guys would define that. Because for example, like the MIT Technology Review, which holds this as an exclusive, that has the word MIT, but like it doesn't get more, academic or scientific than that. Like George Church has come out and, said like we have to kind of take a measured response to that. There have been kind of nuances that I think have been overlooked, right, in terms of like the publicity of industry. I was wondering if you guys could explicitly define what you mean by the response of the scientific community and whether it is even really feasible to have a unified response across the board, considering academic freedoms around the world.
I guess I would say that it's how does science move forward, and it isn't only the responsibility of the scientific community, of course, and there's a wide variety of views, although I think there has been a overwhelming rejection of this particular behavior, except by maybe a few notable exceptions. The question, I think, really is what the communities that are involved in how science moves forward do in a case like this. So it's the scientific community, but it's also the policy community and the state level decision makers, right? So among the things that happened at the moment that we were all sitting in Hong Kong watching this unfold was an interview with Scott Gottlieb, the commissioner of the FDA, saying if the community can't get its act together and control this, then maybe we need to create some hard, concrete restrictions. Right, so there's a dynamic that is part of the answer to that question. So I don't think it's even one community, it's communities.
Yep. And I would add funders and scientific publishers as well.
Absolutely. Institutions.
Yep.
Good question for Jeff. At the Hong Kong meeting, the lay press was universally negative about this. And I'm curious if anybody in the audience or one of the moderators questioned Dr. Head about his choice to circumnavigate these time-tested policies of peer review and so forth. And if so, if he had a response to that.
There was a little bit of that in the question and answer, and a lot of speculation about the motivation. And I think that again, this is talking to lots of people, but we won't know maybe ever. I think his expectation was that he would be lauded as a great innovator, and this was a landmark achievement. And when can I expect the Nobel Prize, right? And this would be marked on a calendar like the first IVF baby, Louise Brown. We all know was born in 1978. It would be like that. Why he had that view, I think, is if that's the right interpretation, of course, is worth a lot of discussion and thinking. So I think the answer to your question is his motivation seemed to be one about being the first rather than one about being the most responsible.
So just following up on the off-target effects piece, so I know that there was some characterization that he claimed to have done on the embryos before implanting. how much characterization do we know about in terms of finding off-target effects? And do we know more broadly if there are guidelines on what level of off-target effects would be acceptable for this sort of thing?
Go ahead.
So, no, I mean, there's been a lot of talk of the need to develop such standards, but we do not have them. And the scientific community, however constituted, has not, been able to establish this is the level that is safe enough. Right, reproduction is a pretty risky endeavor the old-fashioned way. So what is safe enough to be willing to move forward with a pregnancy? As far as what actually was done in the embryos that produced these two kids, or any other pending pregnancy, I don't think we know.
You presented some,
Right.
some, I don't know if data is the right word. He had slides that had some answers to those questions, but the question is what constitutes enough information and what makes something safe. Just because I'm sure this is on people's minds, I think what's going to happen is that there will be lots of work to answer that kind of question, along with lots of others, and these are the steps that need to be taken in a much more concrete model policy, model experimental procedure kind of way, and offering that to state level policymakers for adoption. Because I think the general view is that the level of general principles, as Jeremy articulated, is not enough. We need to give people better tools to help control this kind of research as it moves forward.
And the careful, deliberative way that science was anticipating is to do this in the somatic setting first, where we have some more control, and then potentially move into a more dangerous space where we go. And that was the reason for maintaining that.
With lots of animal work in between.
The question I was wondering about is, if I'm remembering my very recent history right, there was at least one, maybe two instances that were deemed kind of controversial also took place in China in terms of CRISPRing with embryos but not implanted in. And so I have a kind of wide-ranging question, but I'm especially interested since the summit was in Hong Kong where this was announced. If there was discussion about, are his motivations a little more understandable given that there wasn't any kind of, firm decrying at this level when we kind of edged up to the line, right? And if there needs to be a kind of louder conversation about not just hey, this is too fast, too soon, too scary, but we've been kind of messing with this a little bit now, and that takes a bigger conversation about where the values are precisely. Like I said, that was a little vague and wide-ranging, but I'm specifically interested that it was in Hong Kong, that these labs were Chinese labs previously, and was this kind of in the air, if that makes sense.
So the two prior cases, one was in China in the same city, and the other one was in the U.S. that we reported. So very different spaces. But the first one that was reported also similarly in timing, also dramatically positioned, about when it was announced in terms of major international conferences. But the thing about that trial is there was IRB oversight. You may question about whether the IRB has oversight in that setting. They typically don't have many people trained to do that kind of review. But there wasn't the hubris to move ahead without oversight. The second piece was if you were going to do a germline experiment, the mechanisms that they put in place scientifically to prevent any untoward effect were there. Namely, what they did is they selected embryos that had no way of further developing, and they stopped the experiments before they could proceed. And so there was absolutely no way that those embryos would be viable in the first case, and that was the same way. So again, if we were to set out standards for how and when we would do it, it would probably be those. Were people upset that the scientists did it well ahead of the move? Yes, but they put in place some measures that seemed reasonable. I think that's part of what the response was. So I'd be a little cautious to jump to, be careful with your embryos next time you're in China kind of story.
I'd also say there was quite a big response to the initial publication of the work in embryos, and it was a poorly designed experiment, but that's a separate thing. But there was a paper, I can't can't remember the two journals, but there was a paper from Oregon, yeah, Health Science. There was a statement from Francis Collins saying, we're not gonna fund it. There was a rider attached to the FDA funding appropriation bill that prohibited the FDA from even acknowledging receipt of an application to do this. So there was a tremendous response that time. The other thing I'll say is that there are in China, it is my understanding, tremendous incentives and rewards for being first. And the sort of ecosystem of academic scientific research is quite different here.
I don't, Dave, if you want to say something before you have to run, I want to answer Travis's question, but if you want to get in the word.
I'm sorry, I do have to leave, but this has been great, and I think we should view it as an opportunity, because I think the National Academy of Science Committee sort of laid out the general framework, but I think as the point that you made, Jeff, I think it's time to be very specific, and I presume, as the scientific community did with the solar and other examples, that we can do it now. I think there are a lot of scientific details about this that are very fuzzy, but they're less concerning than actually laying out a policy or a plan about how this work should be done.
Yeah.
Not after the fact.
Yeah, no, and I think that's about to happen. There's a lot of work being talked about behind the scenes. I want to speak to the Hong Kong question. So it's instructive not only that the summit was in Hong Kong, but that it was in Hong Kong and not in the mainland. So there's a big difference between Hong Kong and Shanghai, or Hong Kong and Guangzhou, which are 45 minutes apart by car. And there's a reason that the summit was held in Hong Kong and not on the mainland. And the openness to discussion in Hong Kong is very different. So the fact that it was in Hong Kong actually is a positive thing. That said, there is a huge investment, and I think we both kind of, all three now alluded to the fact that China is making major investments in biotechnology. And they want those investments to be respected and thought of as first world and consistent with the rules of the rest of the world. So they're not incentivized to break rules. Now there was an interesting pause between the announcement in the MIT Tech Review and then AP and then the YouTube videos and the statements by the Ministry of Health. There were a number of days that elapsed, and there's some thought that they were waiting to see what the international reaction would be prior to taking a decision, because being first is important. And so the question of whether it was being first in a way that would be lauded or criticized. And the answer was clear, and the reaction to it was also clear, is also clear. So I think that's just part of the story that will be written. There are dissertations here, right? Lots of fodder in the history of medicine world, and STS, and bioethics, and health policy about how this all transpired.
Yeah, so I had a question about the sort of pragmatic approach moving forward.
Sorry, thanks.
So I think, it seems obvious that there's a lot of issues in this current experiment, and it raises a number of ethical concerns that are going to require careful deliberation among bioethicists and also the public at large. And my question is that now that it's been sort of shown that this is a feasible, doable system that is achievable, how can we have those ethical conversations fast enough and widespread enough so that they happen before the eventual adoption of CRISPR system more widely?
Probably all have thoughts about that. Feel free.
Well, and there are lots of parallel conversations we need to be having right now, right? And it is the case that we don't have mechanisms in place to have lots of those conversations, right? I mean, you can do public surveys, but that's not actually going to give you meaningful information about how people feel about this, and it's a one-way system, right? We don't have, like, the UK has a pretty good system of public consultation, although how the outcomes of those consultations get into the policy-making discussion is unclear. And we don't have good, robust systems like that here in the US or more broadly.
Right,
So we have work to do.
Well, we do have, so you mentioned in your your overview about the RAC, the Recombinant DNA Advisory Committee. There's actually a proposal on the table right now, ironically, to dissolve the RAC, or actually change DRAC. But I think changing it may take it was actually re-RAC. It was, the original plan was to simplify the RAC's oversight mechanism, not have it be required and focus more on public policy conversations. What that loses is the ability to learn inductively from the kinds of research that people are proposing. So we can only go so far deductively to say that these ideas and ethical principles we have should play out this way in science. We can get part of the way there. But what we can really learn is to see the actual science that people are trying to do. What are the animal experiments that preceded it? What are the effects, so that we can then develop standards and guidelines that are attuned to the evolving nature of science. So I think this may be a wake-up call for not dissolving the RAC, but actually enlarging its promise to cover all sorts of geography.
Which is actually the proposal, is to make gene transfer research less about 4 times a year review and moving to the model that you're suggesting. I would add one more thing, which is these technologies. So Debra in her presentation had MRT, mitochondrial replacement, listed and didn't talk about it. But the FDA has signaled that MRT and certainly human genome editing would need to go through FDA licensure and approval for use in clinics in the United States. So that's the first time that a reproductive technology would be subject to FDA oversight. So we do have actual policy mechanisms to control the introduction and use of technologies like these. It doesn't speak to the public engagement piece, but we can control it before it gets released to the market in a way that we probably didn't have before, because the technologies weren't deemed to be subject to FDA oversight and approval. Now, the problem with a prohibition like the one that Debra also mentioned is that we then encourage places that have fewer rules to fill the void. So we have MRT clinics in Kiev offering services, which doesn't seem to be the best way for the community to progress, proceed.
All right, so I have a counterfactual question. The situation that we are faced with today was a perfect storm from an ethics and science policy perspective. Everything seems to have been wrong, right?
Yeah.
Everything was violated, everything was wrong. Jeremy had your list of ethical considerations, wrong on every point. This is just fun to, sort of challenging to think about. Let's imagine that the research had proceeded by the scientist in China, and everything had been right except for the absence of oversight. He picked a disease target that would have met the criteria outlined in the national consensus report. The consent had been honest and straightforward. He kept everything under wraps. He'd done the animal experimentation, but he really wanted to be first, and he wanted to come out with a big splash, right? So there's no oversight and there's no transparency, but everything else had been okay. I'm asking the three of you to just sort of suspend reality and imagine whether he would have gotten the response that he hoped he would have gotten, right, or not. So that's my question.
Yeah. So it's rewarding secrecy is sort of your point.
That's my word. Okay.
It's a good question.
I think we'd be in a much more challenging situation than we're in right now. I mean, he made it super easy for us.
That's correct. That's my point.
Yeah.
He delivered a disaster.
Yes, he did. Yeah.
The fact that he didn't appreciate that he was delivering a disaster is itself really interesting.
Yeah.
But what if he hadn't? What if it only been a disaster from the standpoint of failure to disclose, transparency, secrecy, in the interest of being first?
I think the response would have been much more mixed and much more nuanced.
But it seems to me that the major negative that people have been talking about was the fact that he did germline gene therapy. I think he would have still been heavily criticized for doing germline therapy.
Yeah.
Yes, but it's that plus everything else.
Well,
Although I will say, to this point, that the consensus reports, both in the US and the UK, have signaled that germline modifications can be acceptable. And the the UK actually in a more permissive way. They think of the distinction between therapy and enhancement is not a real one. And so they would go further, I think, than the US report did. So I think that there's now a sense among at least people who work on those reports that germline is not the bright line that it happened over time.
It certainly
You can argue about that.
seems to me, looking at the National Academy and National Academy of Medicine comments on it, were very strongly against germline therapy.
Well,
Today.
we can talk about
Today.
that. Yes.
Yeah.
But with a path to go forward in a way that would be responsible and make modifications that would be very germline.
Unfortunately, we are at,
We're finished.
sorry to interrupt, I know.